Okay, class. So here for today's lecture, we will try to look on toward the major fundamental differences that we have in between the eukaryotic and the prokaryotic cells that we have. But before I proceed with this, here you know from the very basic classes and from the prior classes that we have got total of five kingdoms. We have the kingdom Animalia, we have kingdom Plantae, we have kingdom Fungus, and then we have got the kingdom Protoctus. So all the organisms of these kingdoms, they would have these cells in them and they would be the eukaryotic cells. As far as the last kingdom, the kingdom Prokaryotes is concerned, that includes the bacteria in it, so they would have the cells. And these cells, they would not be the eukaryotic, but rather they would be the prokaryotic cells. So we will try to see that what differences lie between the eukaryotic and the prokaryotic cells, and we will see in terms of when they evolved, in terms of the cell volume they have, what kind of the organelles they have in them, what kind of the configuration they have got for their flagellum, whether they would be carrying out the nitrogen fixation or no. Now, for the bacterial cells, this is believed that they are more older or they evolved prior to us, the eukaryotic cells. So the age of the prokaryotic cells, this goes back to around 3.5 billion years ago. So they are the older cells. The eukaryotic cells, they are the newer cells and they have evolved some 1.5 billion years ago. If we try to talk about the size of the cells that we have for the typical prokaryotic and the eukaryotic cells, so the bacterial cell size can range in between 1 to 5 micrometer. But when we talk about the eukaryotic cells, so their cell size can go up to 40 micrometer, and by that they are around 1000 times they have more volume as compared to the prokaryotic cells. And if we were to see the differences in terms of the ribosomes which they have in them, so the bacteria, they strictly have these smaller cementous ribosomes. But when we talk about the eukaryotic cells, so the mitochondria and the chloroplast, whatsoever the organism they would have in them, so they would be cementous ribosomes. And along with that, the ribosomes that we have in the rough endoplasmic reticulum attached to the rough endoplasmic reticulum and the cytoplasm, so they would be the bigger ribosomes and they are the ATS ribosomes. If we try to talk about the organelles, that where differences lie in between the organelles of the prokaryotic and the eukaryotic cells. So the bacteria, they would never ever have any of the organelles that would be membrane bound. Okay, like their nucleus lies naked. Their ribosomes, we know no matter if they are of the eukaryotic or the prokaryotic cells, so they are the non-membrane bound. So this is the case with the prokaryotic cells. Now, for the eukaryotic cells, we will see that the organelles are available within their cytoplasm and they are membrane bound. And by the membrane bound, what we mean is that these cellular structure they could be double membrane. And for the double membrane organelles present in the eukaryotes, we have the nucleus, then we have got this mitochondria, and then we have this chloroplast with us. The eukaryotes also have some single membrane organelles in them, and among them we have the lysosomes. Then we have the vesicles in case of the eukaryotic cells and the large vacuole that we would have in the plant cells, so they have got their unique single membrane outside of them. Likewise, the Golgi apparatus that we have got and the rough endoplasmic reticulum and the smooth endoplasmic reticulum that we have, so they have single membrane boundary around them. And it is important from the MCQ's point of view that like the prokaryotes, the centrioles and the ribosomes that we have got, say they do not have any of the membranes around them. And if we try to be seeing in terms of the differences that we have in the eukaryotes and prokaryotes on the basis of the cell wall, so the bacterial cell wall is made up of the peptidoglycan. And the word peptido mean that their cell wall would have the protein part, and the glycan mean that their cell wall would also have the sugar part in it. Not all the eukaryotes they have the cell walls in them. The algae, the plants, and the fungus they would have the cell wall in them. As far as the cell wall of the algae and the plant is to be concerned, so their cell wall is made up of cellulose. And when we talk about the fungus, so their cell wall is made up of not the cellulose but of another material, and this is chitin with us. Now, if we try to talk about the difference that we have in terms of the flagellum, so the bacteria, those who are motile, those who have an ability to move, so their flagellum have a simple configuration. But when we talk with respect to the flagellum that we have for the eukaryotic cells, so their flagellum have a complex orientation of the microtubule that makes this flagellum. Like we have learned, like you can go back to my previous lecture where I have talked about the configuration of the microtubule that we have in the eukaryotic cells. So it was 9 plus 2 configuration, where we would have two central microtubules, and they were surrounded by 9 duplets of the microtubules. And the flagellum is, in case of the eukaryote, this is membrane bound. This is not the case with the prokaryotic cells. As far as the type of the cell division has to be discussed in this scenario, so the bacteria, they can divide both sexually by conjugation and they can divide asexually by the binary fission. So when we have got the parent bacterial cell and when they divide into two of the new bacterial cells such that the cellular content they remain the same, so it would be the binary fission in a form of asexual reproduction. And when we talk with respect to the sexual reproduction that we have in these bacteria, so this is conjugation. And in the conjugation, the plasmid would be shared between the two bacteria. And as far as the eukaryotes are concerned, so they carry out the mitosis and they carry out the meiosis. They do not have this binary fission or the conjugation situation in them. Further moving onward, for the prokaryotes, some of the bacteria they have an ability to carry the nitrogen fixation. The eukaryotic cells they can never ever carry the nitrogen fixation in them. So here we are done looking on toward the major differences that we have in between the prokaryotic and the eukaryotic cells. So see you in the next class.